The genome of this SARS coronavirus 2 is approximately 30,000 nucleotides long, and to be exact, it is actually 29,903 bases long. But once we amplify the genome of this virus using reverse transcriptase, we do not compare the entire 30,000 long nucleotides, but actually only 100 nucleotide long stretch of its genome is compared because it is specific for this very virus alone. This 100 nucleotide long stretch of its genome has two important genes which are very specific to novel coronavirus. If these two genes are present and if we are able to detect by the PCR after amplification of its genome, if we are able to detect the presence of these two genes, then the test is positive. Then test comes positive. Then the patient is said to be COVID positive. And if only one of the genes is present and another gene is absent, then test is declared as inconclusive. And if neither of the genes is present, then the test is declared as negative. So this is how this PCR used to detect a novel coronavirus. Number second application of PCR is in the diagnosis of specific mutations. We know there are several genetic disorders in human beings which are caused because of the mutant genes. So presence of such faulty genes on DNA can be detected by PCR much before the establishment of the disease. So there are some diseases like Huntington's disease, like cystic fibrosis, like phenylketonuria, like muscular dystrophy, etc. They can be detected or they can be diagnosed much prior to the establishment of the disease. I will here give one example of such disease, that is Huntington's disease. Huntington's disease is caused by the mutation in a gene which is called as huntingtin gene. In normal or non-Huntington individuals, this HD gene has a pattern called trinucleotide repeats. That trinucleotide repeat consists of three bases which are repeated several times, like this C, cytosine, adenine, and guanine. These three bases in normal or non-Huntington individuals, they are repeated less than 30 times. Please note the number is less than 30 times. But in case of the Huntington disease individuals, the individuals having mutant gene, a mutant HD gene, there this CAG trinucleotide occurs more than, please note the word more than 36 times. So if it is present more than 36 times, then the individual will have Huntington's disease, and this Huntington's disease can be detected much earlier before the establishment of disease by using PCR, because PCR that will amplify that gene many times so that we have large quantity of DNA available, then we can sequence it, and if it is and count the number of CAGs, if their number is greater than 36, and then the inpatient will develop Huntington's disease in due course of time. Another important application of PCR is in prenatal diagnosis. So there are certain, again, the genetic disorders that can be detected much before the birth of the individual. So such type of detection is called as prenatal diagnosis. So in case prenatal, there are some diseases which are incurable, then the individuals or the couple are accordingly advised or counseled to abort the child. Another most important function or application rather of the PCR is in DNA fingerprinting. Now DNA fingerprinting, as we know, it has in itself several applications, and nowadays in recent years DNA fingerprinting has become more successful in solving some forensic cases, criminal cases, say for example in solving the murder mysteries, etc. There the DNA sample which is collected at the crime scene can be in the form of a single follicle of hair, and from that follicle of hair, using PCR, we can make millions and billions of copies of DNA that will then be used to identify the actual criminal. Another important application of PCR is in the research. So apart from DNA fingerprinting, in solving the criminal cases or in solving the paternity issues, the same process of this PCR polymerase chain reaction can be used to detect different types of microbes in soil or different types of other organisms in soil, like the soil nematodes for that matter, by comparing them with the already identified the microbes or the nematodes. Another application of PCR is in paleontology. Now, PCR can be used to clone DNA fragments from the mummified remains of the humans and some extinct animals like woolly mammoth and the dinosaurs. The small fragments of DNA which get mummified in the fossils, from that we can obtain the DNA, we can amplify it using this PCR, and we can theoretically clone it. Then the another important application of PCR is in diagnosis of the plant pathogens. PCR technique, it can also be used to detect several plant pathogens. Now plant pathogens can be in the form of virolds, they can be in the form of viruses, tobacco mosaic virus, cauliflower mosaic virus, etc. They can be in the form of mycoplasmas or the fungi or in the nematodes. All these can be detected, their different types can be detected by using the PCR. Hope you have enjoyed this lecture, and with this we come to conclusion of our first topic of this series, that is PCR and its application. Thank you very much for your patient hearing.